In order to grow and divide, cells go through a process of events known as the cell cycle. At the end of the cycle, the single cell splits into two daughter cells. In cells with a nucleus, the contents of the nucleus and the cytoplasm are divided through the mitotic phase, which consists of two portions, mitosis and cytokinesis. Before mitosis begins, the cell goes through interphase, where the DNA is replicated and the cell grows. Mitosis has 5 phases that result in the division of the cell nucleus. The first phase is called prophase, which starts the breakdown of the cell. During this phase, the chromosomes become visible and are paired as sister chromatids. The centrosomes move to opposite poles of the cell and the microtubules extend between them, pushing them farther apart. The nucleolus disappears and the nuclear envelope starts to break down into vesicles. As the envelope separates, the endoplasmic reticulum and Golgi apparatus break and disperse. The next phase is the prometaphase, which is the continuation of many actions started in prophase. This phase ends with the formation of a connection between the cytoskeleton and the chromosomes. More microtubules assemble and stretch across the former nuclear area and form the mitotic spindle. The chromosomes continue to condense and each sister chromatid fastens to the microtubules at the centromere by a protein called kinetochore. The nuclear envelope vanishes. The cell then enters metaphase, where all the chromosomes align in a plane called the metaphase plate. The metaphase plate is midway between the two poles of the cell. The chromosomes are now maximally condensed or raveled, and the sister chromatids are still connected to each other. The next step is anaphase. At the centromere, the sister chromatids are split apart at the metaphase plate. Each chromatid is now a chromosome and is pulled toward the centrosome where the microtubule was attached. The non-kinetochore microtubules slide against each other at the metaphase plate and start to elongate the cell. Lastly, the cell enters telophase, where the chromosomes finally reach the opposite poles and begin to unravel. The mitotic spindles break down into monomers. The monomers are then used to assemble the cytoskeleton for the new daughter cells. New nuclear envelopes form around the chromosomes. Spindle fibers continue to push the poles as it enters cytokinesis. After mitosis is complete, the cell enters cytokinesis, where the cytoplasmic components physically separate into two daughter cells and cell division is completed. Cytokinesis happens differently in cells with cell walls and cells without. In cells without cell walls, cytokinesis happens after anaphase. At the former metaphase plate inside the plasma membrane, a contractile ring composed of actin filaments forms. The actin filaments then pull the cell inward, forming a fissure called the cleavage furrow. As the actin ring contracts, the cleavage furrow deepens until the plasma membrane is cut in two and the two daughter cells separate. In cells with walls, the structure of the wall surrounding the plasma makes a cleavage furrow impossible. Instead, they form a cell plate. During telophase, Golgi vesicles move on microtubules and collect at the metaphase plate. The vesicles then fuse from the center toward the cell walls and form a structure called the cell plate. The cell plate enlarges with more and more vesicles until it merges with the cell wall. A new cell wall of cellulose is then built by enzymes using glucose accumulated between the membrane layers. The Golgi membrane then becomes the plasma membrane in the new daughter cells. That's it for mitosis and cytokinesis. Mitosis can only happen in cells that have a nucleus. That would be cells like eukaryotes, plants, and animal cells, but not prokaryotes like bacteria. Imagine how many eukaryotic cells in your body are going through mitosis this very minute.